I'm interested in how the brain puts together information across the senses. We're all making an assumption. We're assuming that we see the world in the same way, and that's exactly what it is. It's an assumption. Synesthesia is an unusual phenomenon in which people often describe a mixing of the senses. So, for example, a sound might have a visual component. A smell might have a colour or a texture or a form. The most common is where letters and numbers have vivid experiences of colour. It's also intriguing, I think, because it might give us insight into the way we all construct our conscious experience. I want you to smell it and tell me what you see, and I also want you to try and identify.
So it's cinnamon, and cinnamon's, it's kind of all, it's dotty, and it's got pink and black dots and a bit of brown dots, and it's kind of grainy, and it's kind of a high smell. It's kind of up here.
There's a very prominent theory that suggests that attention is the critical factor that binds together information within vision. So, for example, knowing that the colour and a shape of a particular object go together. What our research suggests is that in synesthesia, attention is playing the same role. It's binding the synesthetic experience to the letter or the sound in the same sort of way. There are actually clear differences in how they're using their brains. So different areas are active when synesthetes are viewing letters. I'm interested in synesthesia not just because it's unusual and in and of itself, but of what it might tell us about the way all our brains work, and particularly how we put together all that information into this seamless view of the world that we get.
My expertise is in the repair and regeneration of the nervous system through the nose. We're studying the cells of the olfactory organ, the sense of smell, and this is an accessible part of the nervous system that has a regenerative power. There are stem cells in the nose that regenerate the new nerve cells, so that makes it really interesting. It's interesting because it could give us an insight into how to repair the nervous system, the brain and the spinal cord. About 300 people have a spinal cord injury in Australia every year, and the majority are younger people. Spinal cord injury is thought to be unrepairable, but in the last 10 or 20 years, neuroscience has caught up. Our team was the first team to extract the cells from the nose and to learn how to grow them in culture. Rats with spinal cord injuries had those cells injected into the spinal cord and they regained motor function. All the science is there to show that it could work in humans. So far, our human trial, we've shown that they're safe to put in, so that's the first step. I got into biomedical research because I wanted to make a difference. I think most researchers do want to make a difference. And to see this all coming to fruition, actually going from the lab into the clinic, has been a dream for a long time.
Antarctica is seeing some of the greatest changes on Earth at the moment. And the top predators are often some of the first animals that show change. The first lot of work that we're doing is looking at the leopard seal and looking at changes in the diet of the leopard seal. As a wildlife biologist, I'm really conflicted. In answering our scientific questions, we need to potentially harm the animals that we work on. So it's been important to me to introduce a gentler approach to working with the animals. To collect the whisker samples, we need to go and actually pluck a whisker from live seals. If you cut the whisker up into tiny little segments, each one of those segments reflects the diet at the time the whisker was growing. That gives us about 5 years of diet data from the animal, so it minimises the number of animals that need to be captured. Leopard seals used to be top predators, and they used to eat things like penguins and seals, and today they're not. Leopard seals now in the Western Antarctic are depending almost entirely on krill, and krill is actually a depleted resource. So krill are down by about 80% over the last 30 years. Often those sorts of impacts can cascade and have influences down the food web. So we do work on whales as well. To assess the health of a whale, to collect samples, you needed a dead whale. So what we've done is developed a technique to catch the blow of a whale. It actually holds the important microbial communities, which are really important for things like immunity and those sorts of things. So that's why we can actually collect this important snot. Sorry.
If we're going to regain control of our planet's thermostat, we need to capitalise on natural ecosystems that can capture and store carbon from the atmosphere. The seagrass, they can capture and store carbon for thousands of years, and that makes them so powerful in the fight against climate change. Many people think the seagrasses are facing a global crisis. And what we're concerned about, given that Australia has more seagrass than anywhere else in the world, we have an area much larger than the size of Tasmania. If these systems become degraded, could they leak out that carbon back out into the atmosphere and contribute to this greenhouse gas problem? So my research takes a two-prong approach. The first is understanding factors that give seagrass their resilience. The second thing we need to do is understand the carbon cycle of seagrasses intimately. So we're looking at a number of stresses that are going to happen under climate change. We're going to see changes in rainfall, which will affect their salinity. We're going to see warming waters. We're going to see reduction in light levels as the coastal zone becomes more stormy. So we're in the lab able to manipulate each of those different variables and see how the seagrass respond. I think we've got a long history of not appreciating seagrasses. Not only are seagrasses one of the most powerful carbon sinks on the planet, they support around 50% of the world's fisheries, and also they're very important for a lot of iconic Australian species. Dugongs, turtles, swans, and they're a natural ecosystem, so they're already there, and so it's a matter of looking after what we've got and restoring seagrass from where it's been lost.
It is unquestionably one of the greatest challenges to address is powering Australia in an environmentally sustainable way. And I study alternative energy materials. These are materials that store and release energy as an alternative to things like petrol for the transportation industry. Things like battery materials, which are used in electric vehicles, and an alternative energy technology based on hydrogen. I work at the Australian Nuclear Science and Technology Organisation, which has a nuclear research reactor, which produces neutrons for the experiments that I perform. This is a neutron scattering instrument that I operate. It's called Wombat, and it's the fastest of its kind in the world. It's really spectacular at looking at energy materials. In particular, we get a detailed picture of how these materials function at the very small scale, at the atomic and molecular scale. Neutrons come in and strike this sample, this central part here, and scatter in all directions. And we take a slice of that information, and that gives us a picture of how the atoms and molecules are moving and how they're also arranged. It's not enough to have an incremental advancement in these technologies. What we're really looking for is a discovery, a step change in the advancement of energy technology. And by studying these materials and the way they function at the very small scale, the atomic and molecular scale, this enables the opportunity for such a discovery.
If you take out the essential part of a car, it can't move anymore. That's what we want to do with the malaria parasite. We want to stop malaria in its track. Malaria is the most important parasitic disease of humankind. It affects approximately 250 million people per year, and almost 1 million people die of malaria. Most of those victims are children under the age of 5. Malaria is spread by the bite of a mosquito. Once the parasite is in the body, it infects human red blood cells. They make the infected red blood cells more sticky, and those infected red blood cells then clog up blood vessels. By clogging up blood vessels, it leads to organ failure. I want to find out what molecules of the malaria parasite makes us sick. We discovered a new way of looking at different genes and figure out what the function is. We targeted approximately 100 of them and found that 30 of those genes are actually absolutely essential for the survival of the parasite inside of the human body. And by finding that out, we then have a target where we can design drugs against. Finding essential genes on that scale has never been done before. Science is always an incremental step, so hopefully other people will base their work on my work.